Hi everyone, and welcome to our video on the effect of concentration on enzyme reactions. So this is the 6th in our little enzyme series of videos, and we're looking at that spec statement 2.1.4d, which is just focused on the concentration and how it affects enzyme reactions. First thing we're going to do then is concentrate on substrate concentration. Now, what we actually find is that in these enzyme reactions, if you increase the concentration of the substrate, then we increase the rate of reaction. And we can see that in the little graph in the bottom corner. Then initially, as we can see, as you are increasing our substrate concentration, we're getting the corresponding increase in the rate of reaction. So that's our little diagonal line at the beginning. Reason for that is that we've increased our substrate concentration, therefore more enzyme substrate complexes will form per given unit of time. However, this is not going to continue increasing forever. Eventually, what's going to happen is we reach a maximum rate of reaction, and increasing the substrate concentration is not going to increase the rate. And we can see that where we have the plateau, so the horizontal part of the line on the graph. So at that point, obviously, the rate is no longer increasing even when we've increased the substrate concentration. Why? Because at that point, all of the active sites on any enzyme present are already occupied with substrate. So basically, at any given time, there are no spare active sites going. So because of that, we can have no further collisions between the enzyme active site and the substrate. Therefore, it does not increase the rate any further. The second concentration we're going to concern ourselves with is the concentration of enzyme itself. Now, when we consider the situation for how we change the concentration of enzymes within living cells, then that's all going to be controlled by the rate at which the enzymes are being synthesized and the rate at which they're going to be degraded. So, i.e., how quickly are we creating new enzymes and how quickly are we breaking down the old ones. And the way that this is going to come into play is we're going to have our genes, and hopefully we remember that genes can either be switched on or switched off. So if we switch on a gene for a particular enzyme, then we're going to obviously synthesize it. If we don't want to synthesize that enzyme, we can switch the gene off. So purely by switching those genes on and off, we can control the concentration of enzymes within the cell. It is worth mentioning here that our proteins that we have within the cell are going to be breaking down and then new ones being made from the amino acids that we've then just reformed. And this is really quite advantageous to living organisms, because any abnormally shaped proteins that may have been created are going to be removed. So what we can obviously do is avoid any buildup of abnormal shaped proteins, and we can also regulate metabolism by removing any of those additional enzyme molecules that we don't need. So all we're going to do is break those obviously proteins we no longer need, whether it be just a protein or an enzyme, into their constituent amino acids. In terms of the effect on increasing enzyme concentration, then we see an increase in the rate of reaction as well. Quite a logical thing there, I hope, because what we're seeing is if we've got a greater concentration of enzyme, then there are more active sites available, so more successful collisions will occur. However, we will reach a point where increasing the enzyme concentration is not going to lead to an increase in the rate. Quite simply, we get that little plateau on our graph again. This just comes down to the fact that we get to a stage where there's no additional substrate molecules to occupy those extra active sites. So we have a surplus of active sites, but not enough substrate to fit into them. When we're considering these enzyme reactions, we really should consider this concept of the initial rate of reaction. And in any enzyme reaction, the initial rate of reaction is going to be its fastest rate. And quite simply, the reason for that is this is when we've got the highest probability of a collision between the enzyme and the substrate, because that's the point that those two are in their highest concentrations. As we then progress through this reaction, substrate molecules are being used up. So we're reducing the substrate concentration the longer this reaction goes for. If we've reduced the concentration of our substrate, we're reducing the frequency of collisions, and therefore the rate is going to slow down. So whenever you're looking for that fastest rate of reaction in our enzyme reactions, it will always be the initial rate, so the bit closest to the left-hand side of the graph if you're looking at it. As always, I do suggest you subscribe to the channel so you can see when more videos are uploaded, and head on over to the A-Level Biology website where you can find a range of other resources that are hopefully going to be helpful in your A-Level Biology studies.